A clinical sample may contain many commensal and transient bacteria that do not need to be identified. You wouldn't want to waste time culturing all bacteria if, for example, you suspect a gram-positive infection. Selective media contain chemicals that prevent growth of unwanted bacteria without inhibiting the growth of desired organisms. The swab was inoculated onto tryptic soy agar and phenylethyl alcohol agar. The PEA agar is selective for gram-positive bacteria, while the nutrient agar allowed growth of both gram-positive and gram-negative bacteria. Differential media contain nutrients that allow a technician to quickly distinguish one bacterium from another by how they metabolize the nutrients. For example, pathogenic Staphylococcus aureus can be differentiated from other staphylococci by the enzyme coagulase. Coagulase-positive bacteria reduce tellurite to produce black colonies on tellurite glycine agar. Divide one nutrient agar plate into 4 sections by labeling the bottom. Repeat to mark an MSA plate and an EMB plate. Label one quadrant on each plate for the organisms used. Streak each culture onto its quadrant on the agar. Incubate the plates, inverted, at 35 degrees Celsius for 24 to 48 hours. All 4 bacteria grew on the nutrient agar. Note the pigments produced by 2 of the bacteria. Pigments are useful identifying traits. Gram stains are used to characterize each organism. Unfortunately, most gram-positive cocci look alike and most gram-negative rods look alike through the microscope, so you don't have much identifying information yet. Mannitol salt agar contains peptone, mannitol, and 7.5% sodium chloride and phenol red. The medium selects bacteria that can tolerate the high salt concentration. Organisms 1 and 2 were able to grow in our example. Phenol red is a pH indicator. Bacteria that ferment mannitol produce acid, which turns the phenol red yellow. Eosin methylene blue agar contains peptone, lactose, sucrose, and eosin and methylene blue dyes. The dyes select gram-negative bacteria, and the sugars will differentiate lactose and sucrose fermenters from non-fermenters. Bacteria that ferment one or both sugars will have dark centers. Escherichia, shown here, is almost black. It shows a green metallic sheen in reflected light. Another fermenter, Enterobacter, has dark colonies with a light border. Non-fermenters will have colorless colonies. You will be able to determine which medium selects gram-positive and which selects gram-negative bacteria. Thus, you will learn which medium to use for a skin infection or for an intestinal infection. Dispose of the plates properly.